Plain cage. Or empty cage. White cage. Okay, good evening. Say good evening. Yeah. I asked you to bring your study material. Forgot? Yes. What hurry? You remember? At least now? Okay, fine. And actually, you are supposed to write your test bed still. If people ask for some time, right? Yeah. The whole chapter finished. Vijay Lakshmi, right? The whole chapter actually finished. For you, two people only like revised last class. Yes, you remember all those things. So please read your text also and your study material also. Okay, fine. So today we are just going to see the structure of something called actually genetics. That's what we studied in like the last few classes. Now it is actually called what? Molecular genetics. Okay, what is called molecular genetics? Okay, can you see that? So it's called molecular genetics. See, what is genetics then? Genetics, the study of inheritance and variation. The study of genetics is what? You forget all those things? Forget. Happily forget. Okay, see, what is evolution then? Origin of earth, first life, and all life forms. Correct? That's evolution. Okay, evolution has three components. What are the three components? Heredity, variation, and then natural selection. Is that no? What is heredity? Inheritance of characters from, yeah, inheritance of character. Yes, from parents to progeny. Variation means how, yeah, progeny varies from, differs from parents. Okay, how progeny differs actually, differs from parents. Natural selection talks about which organism is having more survival, the adaptation. Okay, adaptation, existence, and then extinction, everything. Okay, so like that, how different organisms are existing and why many organisms are extincted. That's what actually like. And then in heredity and variation only we studied about the rules of heredity, right? Rules, Mendelian laws, monohybrid cross, dihybrid cross, all these things we discussed. Okay, so that is called coming to genetics. There are three parts. What are they? Classical, or you can say transmission genetics. What is transmission genetics? The classical genetics, how characters are transferred from parents to progeny. That is called the transmission genetics, study of how characters, like Mendelian experiments, all these things, how characters transferred from parents to progeny. That's transmission genetics. And second one is actually called what? Molecular genetics. Okay, molecular genetics. What is molecular genetics? Study about the molecules which is responsible for inheritance. Molecules are responsible for inheritance. Okay, that is molecular genetics. And what molecules you are going to know? You should know about DNA, and then chromosomes, genes. Yes or no? You should know about in molecular genetics, we will be studying about the DNA, chromosomes, genes. Okay, fine. So, based on this, the material which is given, know about these terms and other important things. So, that's the main thing about today's class. And tomorrow, definitely bring your material. We'll have and go through all the entire chapter, and we will have a common discussion. Okay, I think thereafter we don't have to spend too much of time with this particular chapter. Okay, long time we took actually for this chapter. Okay, fine, good. So now, who discovered chromosomes? Strasburger. His name is what? Chromosomes. Chromosomes. Did we discuss about what is DNA, what is chromosome, what is gene already? You know about that? What is DNA? Hmm? Hmm? Yes, come on. Okay, fine. Where DNA is present in our body? Hmm? Yes, generally, apart from the textbook informations, where DNA is present in your body? Hmm? Nucleus. Where nucleus is present? Where cells are present? All of the body is made up of cells only. We are nothing but cells, group of cells, different type of cells. Okay, fine. So, if this is the cell, and in the nucleus, like this, the chromosomes are present. Okay, fine. See, coming to the important facts about the DNA. Okay, so here you can see the structure of DNA. Can you see the salient features of? Is it clear? The salient features of the structure of DNA. Okay, fine. So let's go in reverse order. Then that helps us to understand what DNA, what chromosome, and what are genes. Okay. So who discovered DNA? The double helical structure of DNA. Watson and Crick proposed this model. Okay, since it is there in your learning material, we are just discussing about this. Okay, so DNA, like it is a double-stranded molecule. What it is a? It is a double-stranded molecule. Okay, molecule It is present in every cell. What is the length of the DNA? Yeah, we discussed, right? Do you remember? It is 2.2 meter. Okay, 2.2 meter DNA length. Okay, it is packed into chromosomes and it is not present in a continuous manner. You remember, I think I told you all this is. Okay, DNA is a linear structure. It is double stranded and it is present in a discontinuous manner. Okay, segmented actually. Okay, not present in a continuous manner. It is actually present in different pieces. These segments are only called what? Chromosomes. Okay, these segments only are called what? Chromosomes. Okay, fine. Chromosomes. Why they are called chromosomes? Normally in a cell, the DNA is not like it is present in scattered throughout the cell. You cannot see. Only through electron microscope you can see. In normal microscope you cannot see. Okay, because it is kind of dissolved form. It is present, occupies the entire nucleus. We cannot see. But only one time we can see. When? During cell division process. When you can see DNA? Cell division process. That time only we can see DNA, because chromosomes. Because like this, the chromosomes are present normally when it is observed under microscope, but during the cell division process, especially mitosis, there are four phases of mitosis. Okay, in higher class you study about I think in class 9 you would have studied about cell division. You remember? I think last part of your cell is cell division process. Okay, mitosis. So there four phases: prophase, metaphase, anaphase, and then telophase. Okay, these are the four phases. And here in metaphase only, like if this is the cell, the chromosomes are aligned in the metaplate. Okay, like this. Okay, that time only they get this structure. And this is the basic structure of chromosomes. Can you see? This is the basic structure of chromosomes. Like this. Okay, since it is homologous and one chromosome you get from Appa and one from Amma, you get this structure, X-shaped structure. Can you understand? So this structure you get for chromosomes. Otherwise, it is present like this. Which we cannot see. Okay, like this only it is present throughout the nucleus. Okay, this is nucleus. Actually, in cell division, you cannot see the nucleus. It is disappeared. Nucleus is disappeared. Entire cell is occupied by chromosomes. Okay, so DNA is linear. We cannot see only during the cell division process. That too, this metaphase. In that phase only, DNA is what? Condensed. To form what? DNA is condensed to form what? Chromosomes. Why? Again, chromo means what? Color. Chromo, color. Okay, so you can add some dye. Okay, you can add some dye. That time you can add some dye which is positively charged. The dye will go and bind with the negatively charged DNA molecule, so it produces a color. Okay, by that color, the dye is actually called what? Acetocarmine. What is that dye called? Acetocarmine or acetoarysin. In cell, if they add this dye, only during cell division process, only during mitosis and metaphase stage, that time this dye will go and bind with the DNA. So now you can give color to the DNA and see. Okay, so that's why it is called only giving color when it is in condensed form. That's why it is called what? Chromosome. Okay, and chromosomes are the carriers of genes. Okay, through chromosomes only, the genes are carried from Appa to the maybe offspring. Appa and Amma to the offspring. Can you understand? Fine. So you should know about the basic difference between DNA, chromosomes, genes. Okay, gene is a single segment. DNA is a linear long one, condensed form piece is chromosome, and then chromosome contains hundreds and thousands of genes. Is it clear? Fine. So here the double helical structure, the model of DNA is actually produced. What it says, the salient features of double helical structure, Watson and Crick. Okay, for the scientists who proposed a double helical structure, Crick. You can say Watson and Francis Crick. James Watson, James Watson and Francis Crick. They proposed double helical structure of DNA. So how this DNA looks like, they are telling. Yeah. Okay, like this. Can you understand? So such kind of or helical structure. Okay, the DNA. Is it clear? I'll make it little more bigger, just for your clarification. This is one star, and it comes like this. Can you see? The double helical structure is like this actually. Okay, two strands are coiled like a ribbon. When you turn your ribbon, coil your ribbon, it gets such kind of structure. See what Watson and Crick proposing. This width, length is what? Length, what did we say? 2.2 meter length. Okay, this width, how much? The width, it is 2 nanometer. What is the width? 2 nanometer. And then this is actually called what? 1 ton. See, you can see this one, and this is called 1 ton. You can see one bend like this, one bend like this, and one bend like this. This is 1 turn actually. They calling it as 1 turn. That 1 turn consists of what? Approximately 10 base pairs. That 1 ton means here 1, 2. Let me use a different color. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Can you see? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Can you understand? So 1 turn consists of what? 10 base pair. Actually 10.5, you just remember 10 base pair. This length is actually called what? The 10 base pair length is actually called what? Size is length of 10 base pair equal to 3.4 nanometer. Okay, then each base pair, length of each base pair equal to how much? If 3.8 divided by 10, it is 0.34 nanometer. Can you understand? So this distance, how much this distance is? 0.34 nanometer. The entire distance is actually what? 3.4 nanometer. One turn. It is called one turn. One turn equal to 3.4 nanometer. Can you understand? And so DNA is a polymer. Okay, DNA is this is the basic structure. Now DNA is a polymer. Polymer means what? Macromolecule, bigger molecule. Many monomeric units are there. So DNA is a polymer, consists of many monomers. Monomers. What are these monomers are called what? Nucleotides. Monomers are called what? Nucleotides. DNA is a polymer which is made up of what? Monomer called nucleotides. Nucleotide consists of what? Three basic units. Okay, that monomer nucleotide consists of what? Three basic units. Okay, here maybe in your yellow material you can see that the three basic unit, okay, called what? One nitrogen base plus one 5-carbon sugar plus phosphate group. What are the three components? Nitrogenous base. You can say nitrogenous base plus sugar plus phosphate. Okay. All three consist of what? Makes a nucleotide. Okay. Now, this nitrogenous bases are classified into two types called what? Purines and pyrimidines. Okay, purines and pyrimidines. What are called purines? There are two molecules, adenine, guanine. Okay, adenine and guanine are purines. Thymine, cytosine or pyrimidines. Thymine and cytosines are what? Pyrimidines. Okay, structurally they are classified into. It is abbreviated as what? Adenine is abbreviated as A. Thymine is abbreviated as T. Guanine is abbreviated as G. Cytosine is abbreviated as C. So A, T, G, C. All these things are what? Nitrogenous bases. Okay, fine. Now, the sugar DNA is made up of 5-carbon sugar. This is the basic structure of a sugar. Okay, 5-carbon sugar. This is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and here carbon 5. There are 5 carbons are there. Pentose sugar it is. See here to the first position, and this is sugar, and to the first position nitrogenous bases are added. For example, adenine is added. Okay, this is first carbon atom, and then to the fifth carbon atom phosphate group is added. Can you see? You have nitrogenous base, you have sugar, and then phosphate. Okay, this is one nucleotide. This is a monomer nucleotide. Okay, like that, by different bondings, many monomers are connected together and DNA is formed. Can you understand? So basic structural details you should know. And then I told you, if only sugar plus nitrogenous base is there, it is called what? Nucleoside. It is called what? Nucleoside. What is nucleoside? Only sugar and nitrogenous base, no phosphate molecule. If phosphate also there, then it is called what? If also phosphate is there, then it is called what? You know, nucleotide. Can you understand? Nucleoside and nucleotide makes the monomer nucleotide. Okay, fine. So why you should know about all these things? What is DNA? It's a polymer. It is made up of nucleotides. What are nucleotides? Made up of these three simple units: nitrogenous base, sugar, and phosphate. So like that, it is a very big chemical thing, but little you please know. At least these things you should know. Okay, basic structure of DNA molecule, the length, the width, all these things. Is it clear? And here the size is actually I gave you in nanometer, but here it is actually given in Armstrong. Okay, little conversion is needed, Armstrong. Okay, so and one more thing you should know is something called what? Base pairing rule. What is called base pairing rule? See, we saw adenine, right? Adenine always bonds with what? Thymine. So it is called what? A double bond T. And then guanine bonds with cytosine. So three hydrogen bonds. What is the bond which makes these two bases? Hydrogen bond. Can you understand? So adenine bonds with thymine with two hydrogen bond. Guanine bonds with cytosine with three hydrogen bonds. So this rule is called what? Base pairing rule, proposed by Watson and Crick. Okay, Watson and Crick. And then another rule is actually called what? Chargaff rule. Just Chargaff's rule. What it says? Purines equals pyrimidines. What are purines? What are purines? We talked two molecules right there. Adenine and guanine are purines. Thymine and cytosine are pyrimidines. Do you remember here? We saw adenine and guanine are purines. Thymine and cytosine are pyrimidines. So adenine cannot bond with guanine. Thymine cannot bond with cytosine. The structure will not allow. Only adenine bonds with thymine with two hydrogen bonds. Guanine can bond with cytosine with three hydrogen bonds. Okay, this is base pairing rule, and Chargaff rule is purine, adenine plus guanine. How much purines are there? Which equals what? That much of thymine plus cytosine. Can you understand? If Can you tell me if 100 A, how much T? If 100 adenine are there, how many thymine will be there? Very good, 100 thymine. Okay, if 200 G is there, how many C? 200, because that's the only option. Can you understand? So this is Chargaff, scientist rule. Is it clear? So all these things make the double helical structure of DNA. Is it clear? Fine. So you should know about DNA. And then coming to the chromosomes. Okay, look at that. This is the structure we get for chromosomes. Okay, what is the structure? See, listen. One arm. Here you can see. Okay, this is called what? Centromere. The center constriction is called what? Centromere. Central constriction is called what? Centromere. When DNA is condensed, it gets this structure. You can have what? Two different arms. Okay, here one arm one, arm two. Sometimes both the arms are of equal size. Such kind of chromosomes are called what? Metacentric chromosome. Okay, when the centromere is present in the between, then it is called what? Metacentric chromosome. Sometimes if the chromosome is having this structure, what is the difference between these two structure? One short arm, one long arm. Short arm and long arm. This structure is called what? Submetacentric chromosome. Basic length. Say we have humans, we have 46 chromosomes. Each and every one will have a different length, and it is not a permanent structure. Okay, it is a temporary structure. During the cell division, it comes. I will tell you this width first. It's actually called what? How much? It is 700 nanometer. Okay, this is 700 nanometer. And this length varies actually from maybe 1000 nanometer to, you can say, micrometer. Micrometer, 1 micrometer. Can you understand? 1000 nanometer, because one chromosome, for example, your Y chromosome is this much size only. Human Y chromosome is the smallest chromosome. This is very small. But chromosome number 1 and X chromosomes are bigger chromosomes. Can you understand? Micrometer la they may be present. Is it clear? Depends. Okay, because it is a transition state. At one particular state only it comes like this and goes like this. Always it is there. Is it clear? Fine. So basic structure of chromosomes. Centromere, two different arms will be present. Is it clear? Fine. Structure of a chromosome you should know. And here you can see. Okay, can you see? Metacentric, submetacentric, and then very short arm, very long arm. Then that is called what? Acrocentric chromosome. Acrocentric chromosome. And then you can't even see the other arm. Only one arm you can see. Then it is called what? Telocentric chromosome. The structure is here. Okay. Metacentric, submetacentric, acrocentric, and telocentric chromosomes. Okay, this structure, chromosome structure. Only during cell division, the first one is normal chromosome, like this, it is present. And during the cell division only, if this is the chromosome, it produces its copy. Can you understand? This is called what? Sister chromatid. I think we discussed all these things. Sister chromatid. Can you understand? So chromosomes, chromatids, that you should know. Okay, yeah? Fine. Okay, how many chromosomes we have? We also studied about sex determination. How many chromosomes we have? Yeah, so 23 pairs. Okay, so 22 are autosomes, 22 pairs. One pair is, you can say, allosome or sex chromosome. Allosome or sex chromosome. Is it clear? Fine. So what is our sex chromosome? You remember? X and Y. X chromosome, Y chromosome. There are two types. In male it is heterogamety. Female is homogamy. Can you understand? So chromosomes, there are number of chromosomes you should know. What is the symbol for chromosome number? It is N. Is it clear? It is what? N. And then what are the common properties of chromosomes and DNA? Properties. You know what are chromosomes? The basic discovery, the structure of chromosome, types of chromosomes. Okay, condensed form of DNA is chromosome. Okay, condensed form of DNA is chromosome. And then the structure, you can get that arm-like structure. Okay, and then types, what are the 4 types? Metacentric, submetacentric, acrocentric, telocentric. Position of the centromere, that's one thing. And other than that, autosomes, allosomes, body chromosomes, sex chromosomes. Is it clear? So these things you should know. And now coming to the properties of chromosomes. What are the properties? Okay, very tough, right? What are the properties? Chromosomes. See, if you have one pen, can it make another pen? Okay, only living cell can produce its own copy. Can a chemical substances can make its copy? One chemical can make another chemical. Is there any chemical? Only one chemical. So that's the chromosome. Okay, means the DNA. Only DNA can make its copy. No other molecule can able to make its copy. So that process is called what? DNA or chromosome to another DNA makes that copy. This process is called what? One DNA produces 2 copies actually. That process they call it as what? Replication. Have you heard about this term? Replica. Replica means what? Exact copy. Okay, so one chromosome can make another chromosome. So that property is actually called what? Important property called what? Replication. Okay, and another one is called what? Transcription. What is called transcription? See, this is DNA, which is double stranded. DNA double stranded. DNA can make what? One single stranded RNA. From DNA, what can be made? RNA can be made. Okay, from DNA can make its own DNA. That process, what did we say? Replication. From DNA, if RNA is formed, that process they call it as what? Transcription. What is transcription? From DNA to RNA. The formation of RNA from DNA. That is called what? Transcription. Is it clear? So DNA can make RNA. From RNA, what can be made? Proteins. From RNA, what can be made? Protein can be made. This process, they call it as what? Translation. This process, they call it as what? Translation. DNA to DNA is replication. DNA to RNA is transcription. RNA to protein is translation. So they call this concept as what? Central dogma. What is central dogma? This process, DNA, RNA, protein. Central dogma is what? From DNA, RNA is made. From RNA, protein is made. Can you understand? This process is called what? Central dogma. Without this process, a cell cannot divide, a cell cannot function, nothing will happen inside the cell. Is it clear? So that you should know. And apart from that, the common something, what is mutation? DNA damage. When DNA can replicate, that time any error can come. So that results in, if the error is not repaired properly, it results in what? Damage, mutation. Can you understand? So that part you should know. Chromosome, DNA, they can make its own copy. Error comes, it replicates, means it rectifies the error. So damage and repair processes. So chromosome and DNA are able to repair the damages. Damages can come, but they can repair. Is it clear? Fine. And then apart from the other functions, heredity informations are carried through chromosomes. Okay, cell can divide only if nucleus divides, only if the chromosome replicates. Okay, mutation. Sometimes because of mutation only new organisms are formed. Okay, and then chromosomes like DNA together helps in continuity of a life form. So all common functions you should know about what? The chromosomes. Is it clear? So DNA, chromosomes, all these things you should know. Okay. So now the last for 5-10 minutes for your doubt clarifications. If you have this material only, it is easy for you to understand, but still. Okay. So go through the material. If you cannot understand anything, please ask me. Okay, that is actually extra material, not much questions for board will come, but little they will touch about the structure of DNA, all these things. So on, if you have some self-interest only, you can understand this part. Okay, like it's a very big thing, but I just wanted to give one concise information. Okay, but DNA, about DNA, all its property, chromosomes, genes, these things you should know. Okay, you must know. Okay, so now we just go through your book. If anything from genetics and evolution we missed, just check that alone. And tomorrow definitely bring this book and get all your doubts clarified. Okay, and because next time question paper will be set for your assignment and all. Okay, assignment, test, everything. But this week I think you have your test in what chapter? Reproduction, the previous chapter. Okay, so and there you focus much on reproduction in flowering plants. So most of the questions are asked from that only. And I don't know how much we covered. We might have missed, but still, hmm? Yeah, 40 marks. So please go through that. Like your material, most of the content from this material will detailed the most plants. Okay, so plant reproduction, flower odor components. Okay, types of androecium, gynoecium, all these things very much in detail. Okay, like a lot of terms you would have not even heard about those terms, but they all will come. What are you listening to the term? Okay, you would have not even discussed or heard about it, but such kind of terms are there. So that, yeah, that the idea is at least you should know the questions. Okay, but if you read this book only, you can answer that. Okay, Kaushik, any doubt, Kaushik? Hello, Kaushik. Kaushik, are you there?
Yes, sir.
Yes. Any doubt, Kaushik? Definitely you have actually. So please go through your study material very well. Note down your doubts and tomorrow you can get it clarified.
Oh,
Okay?
sure.
Yeah. Please read that material.
Okay.
Okay, can I ask some simple questions? Can I ask some simple questions? Yes. What is DNA? What is DNA? DNA stands for what? Deoxyribonucleic acid. Deoxyribonucleic acid. Okay. DNA is made up of what kind of monomer? Monomer. You hurry. Monomer of DNA. DNA is a polymer made up of what? Nucleotides. Nucleotides. Nucleotide consists of what? At least now you tell me. There are three components in nucleotide. Hmm. Hmm. Phosphate group. Okay? Phosphate group. Okay, phosphate group. Actually, that's a phosphoric acid. Okay, fine. And then purines. Adenine. Adenine and guanine. Pyrimidines. Thymine and cytosine. Cytosine. Thymine and cytosine. They are the pyrimidines. Okay. And Rohit, what's base pairing role? Adenine bonds with thymine with two hydrogen bonds, double bond. Guanine bonds with thymine, cytosine with guanine, cytosine. How many hydrogen bonds? Three hydrogen bonds. That's base pairing rule. A plus G equal to T plus A is what? Purine equals pyrimidine. In the rule, is it? Chargaff rule. Okay, Chargaff rule. Okay, fine. So anyway, with this, let me stop. And next class tomorrow is vector DNA. See, listen. In bacteria, plasmids are there. Okay, in bacteria, they sare poranarada ni anda kukta. Okay, see, listen. In bacteria, this is bacteria. Here DNA is there. That's called what? Chromosomal DNA. DNA. And then here also bacteria will have a double stranded circular DNA. They are called what? Plasmid DNA. They are called what? Plasmid DNA. Plasmids, how can you define? They are double stranded. You can see two rows, 1, 2. That's a double stranded. Circular. They can replicate themselves. One can divide into two. And then two. That's called what? Self-replicating. Self-replicating. This is chromosomal DNA. This is extra chromosomal DNA. Extra chromosomal. Okay, DNA. That is called double stranded circular self-replicating extra chromosomal DNA, which can carry the genes from one bacteria to through plasmids. Gene can read from one bacteria to other bacteria. That's why since they are the carrier, they are called what? Vector. They can carry, transfer, gene transfer. They can transfer genes from one bacteria to other bacteria. Is it clear? That's why they are called what? Vector, vehicles. Okay, yeah? Fine. Anyway, so let me stop. Take care. Thank you. Kaushik already left. Okay, thank you. Thank you. Ari, you're not at all serious. Mm.